Hi, my name is Parag Meswani, and I'm the Chief Commercial Officer and Head of Business Development for Sio Gene Therapies. Thank you to the meeting organizers for the opportunity to discuss this company and the work that our employees are doing to liberate patients from severe genetic diseases. It's really a pleasure to provide an update on our pipeline and our portfolio as part of this presentation. Sio is a publicly traded company under the ticker SIOX on NASDAQ. And so as part of this presentation, I may be making forward-looking statements. First, let me start off with some background about Sio and some key highlights about the company and our pipeline. First, we're a company that has 3 clinical stage programs. And we believe that the pipeline that we built over the past couple of years has balance. What do I mean by balance? Well, I mean that we have a pipeline that leverages both traditional applications of AAV-based gene therapy in monogenic diseases like GM1 and GM2 Gangliosidosis, but we're also extending the reach and the promise and the potential of gene therapy into more prevalent indications like Parkinson's disease, where we're using an in vivo lentiviral vector system. Across each of these programs, we believe that we have a leading development position. All programs have potential best-in-class and first-to-market profiles in their respective indications, and they're all in the clinic, and they're all beginning to generate data. We're quite excited about what the future holds for each indication and for each patient population. And we're doing this on the basis of a strong foundation with a strong and capable team and a strong balance sheet that will allow us to get through key value inflection points for each program over the course of the next year and beyond. There's a lot of companies in the cell and gene therapy space, and one of the questions that we get asked often, and one that we're proud to answer, is what makes Sio Gene Therapies different? What we've begun to do over the course of the past 2 years is to build a unique expertise in developing CNS-targeted gene therapies. And our whole philosophy here is product indication fit. We're not beholden to a given vector serotype or a given route of administration. What we strive to do is design our vectors, to design our gene therapies, and to develop a clinical development program that is designed to optimize for product indication fit to give patients the best chance at a response and at success. So what we've been building are unique CNS delivery capabilities, where we have programs that have targeted CNS delivery into the deep brain structures and into the cisterna magna. That's in the setting of our GM2 Gangliosidosis program. But we're also not afraid to employ a systemic delivery if the disease warrants it. And that's the path that we've chosen for our GM1 Gangliosidosis program. And as I mentioned at the outset, we have balance in our pipeline, and we strive to be modality agnostic, where we use both lentiviral vector systems and AAV-based capsids to meet the needs of each indication. And we've generated data based on well-validated targets that have been established in large animal models of disease. That holds true across each of these 3 indications. From a manufacturing perspective, we have a hybrid manufacturing strategy where we have a team that is employing and executing in-house analytical development work for our pipeline. But we're partnered with leading manufacturing organizations where we're able to learn from them and advance our programs in partnership with them to each next stage in development. These include organizations like ViralGen, that is part of AskBio and now Bayer, who supports us in our AAV program, and Oxford Biomedica, who is our partner on our Parkinson's program using their capabilities in lentiviral vector manufacturing. And so this is our pipeline of 3 clinical stage in vivo gene therapy programs. The candidate that is furthest along is AXO-AAV-GM1. This is an AAV9-based program that delivers the GLB1 transgene for the treatment of GM1 Gangliosidosis. This is in an ongoing phase 1/2 dose escalation study that's being run out of the NIH, where we've already begun to generate data at the 6-month mark. And we're quite excited that in the month of October, we expect to share 12-month durability data, looking at safety, efficacy, and biomarkers from our low-dose cohort. And that'll set the stage for a conversation we expect to have with regulators in 2022 to define the path to the market into registration. We have AXO-AAV-GM2, which is our second AAV-based program In this setting, we're using the AAVrh8 vector to deliver 2 transgenes that are involved in the formation of the beta-hexa enzyme in the setting of GM2 gangliosidosis. This is a really special program in a disease that is quite difficult with high morbidity and mortality. And we're pleased to have an open IND where throughout this calendar year, we have and will continue to screen and enroll additional children with GM2 gangliosidosis as part of our clinical development plan and study that's being run out of Mass General Hospital and UMass Medical School. And then finally, we have AXO-LentiPD, our lentiviral vector program for advanced Parkinson's disease. We've begun to generate data in an open label setting that shows the ability to improve motor function and quality of life. And this year, our focus has been on manufacturing scale up. And in the fourth quarter of this year, we expect to have QP or qualified person certification of clinical trial material that will allow us to resume our clinical program in calendar year 2022. So let me give you a quick tour of each of these programs, starting first with the rare fatal pediatric diseases of GM1 and GM2 gangliosidosis. For GM1, this is a really important slide. GM1 is a disease that we've begun to understand very deeply. And this is a progressive pediatric lysosomal storage disease with multi-system manifestations and unfortunately no improved therapies. And our goal here is to tackle this disease at its root, which is we know that there is a deficiency in the beta-gal enzyme. The consequence of that deficiency is the accumulation of toxic substrates like GM1 ganglioside. And you can measure GM1 ganglioside across multiple organs, like the brain, like the heart, like the eye. And our goal here is to deliver the GLB1 gene to increase beta-gal enzyme activity, but more importantly, to decrease the accumulation of these toxic metabolites that lead to increasing morbidity and increasing mortality, and ultimately tackle the multi-system manifestations of this disease. This next slide in very stark terms illustrates the natural history of infantile versions of both GM1 and GM2 gangliosidosis that shows the rapidly progressive nature and uniformly fatal nature of both of these diseases, where there are characteristic hallmarks, like the inability to attain and retain normal developmental milestones, like the ability to transfer objects from one hand to another, the ability to maintain head control, among many others. And so we're trying to tackle and address the natural history of this disease, which we know is a predictable age-related decline in functionality that will progress as children age. So our approach with GM1 is to leverage the years of research that have been going on, both for AAV9 vectors, but also for GM1 gangliosidosis. We know that the AAV9 vector system has been shown to cross the blood-brain barrier, allowing for meaningful transduction of both the CNS and peripheral tissues in GM1 mouse and feline models. The benefit that's been observed here is broad transduction, along with cross-correction of neighboring cells that has the potential to address the multi-system manifestations of this disease. And indeed, we've seen data in naturally occurring models of disease, large animal models like feline models, to demonstrate restoration of wild-type survival. And so our goal in the clinic is to recapitulate those outcomes. And as I mentioned earlier, we're already in the clinic now, where as of September of this year, we've treated 9 children across our low and high dose cohort of AXO-AAV-GM1. We've completed targeted enrollment of type 2 patients in both the low and the high dose, and we've already treated the first child with type 1 or early infantile disease, which is the most severe variant of this disease. Data is being generated. We've shown the ability and proof of concept that you can improve biomarkers of disease severity and disease activity, and preliminary clinical outcomes that demonstrate early signs of disease stability. So we're really excited about where we are with this program. And as we look ahead to our clinical development map here, we're making great progress. We're currently in stage 1, which is the dose ranging portion of the program. And as I mentioned, we've treated 8 patients across the low and the high dose in type 2 children with GM1 gangliosidosis. And we're in the midst of continuing to enroll more children in both the low and the high dose of type 1 or early infantile onset disease. Once we complete the dose ranging series, our goal is to take that safe and efficacious dose into stage 2, which we view as more of the registrational component of the program, and drive that forward to understand safety and tolerability over time, evaluate markers of disease progression in both the serum and in the CSF, like GM1 ganglioside, and look at developmental changes in disease progression over time. And as I mentioned, we've already begun to generate a look at this data from our low dose at 6 months, where in summary, we've seen a clean safety and tolerability profile. We've observed sustained GM1 ganglioside reductions in the CSF. So with systemic delivery, we've been able to demonstrate at 6 months that you can get into the CSF and reduce this toxic metabolite in GM1 ganglioside. And we observed that in 4 out of the 5 patients during the observation period. And all patients showed signs of disease stability across multiple measures of neurodevelopment, like communication, activities of daily living, as well as motor function. So this is at the 6-month mark. And so we're quite excited to extend this data to look at durability at the 1-year mark in October of this year. And as we look ahead towards generating additional data for this program, there are just foundational activities that we've begun to put into place to support the future commercialization of this program. These include really critical things like partnering with patient advocacy organizations like the Cure GM1 Foundation and NTSAD, to setting forward a clear and coherent manufacturing strategy for sufficient clinical and commercial supply through our partnership with ViralGen, through supporting patient identification and engagement initiatives with parties like Invitae, Global Genes, and Screen Plus, which we know is a requisite in rare diseases like this, which is driving increased disease diagnosis and increased disease awareness. Moving on to our GM2 program for Tay-Sachs and Stanhope disease. In this setting, our approach is one-time delivery of 2 vectors using the AAV-RH8 vector that encodes for both the HEX-A and the HEX-B genes. And the thesis here is to go direct to the CNS. Both Tay-Sachs and Stanhope are much more of a CNS manifested disease. And so the thinking here is through a dual intrathalamic and intrathecal route of administration. This will allow for transduction across the entire neuraxis to address neurologic manifestations. And indeed, we've tested this approach in large animal models as well, feline and sheep models, where we've seen an improvement in the clinical rating score and survival extension. As of late last year, we now have an open IND, and we've begun to execute our clinical study plan, which you can see illustrated on this slide here. Very similar in approach to what we've done with GM1, where we're starting with stage 1, which is our dose ranging portion of the program to identify the safe and efficacious dose. And then the plan will be to move into stage 2, which is the more registrational component of the program. We've already treated a child at our starting dose. I've treated a second child at our low dose. And for this calendar year, our focus is on continued screening and enrollment in this particular study. And finally, let me move on to our Parkinson's program, AXL-NT-PD. So AXL-NT-PD is targeting PD, which, suffice it to say, is a very large market, but more importantly, is a market that is still underserved, where half of patients with PD will develop motor problems within 5 years of diagnosis, despite standard of care. And while standard of care treatments like levodopa and dopamine agonists are effective in the earliest stages of the disease, we know that they wane in efficacy over time, where there is an increasing pill burden and an increasing side effect profile that leaves room to be desired, and a substantial opportunity to improve motor function and quality of life. With AXL-NT-PD, our goal is to deliver the 3 transgenes that are involved in the body's production of dopamine, which is that deficient biochemical that leads to Parkinsonian symptoms. The thesis here is to develop a dopamine biofactory directly within the CNS, leveraging the larger packaging capacity of the lentiviral vector system that is delivered through a stereotactic delivery directly into the putamen using neuroimaging with MRI. So essentially trying to localize the gene therapy to the right part of the body that is required to create dopamine in vivo. This is a program that is also in the clinic, where we're beginning to generate data in an open label setting that gives us quite a bit of excitement around patient benefit, where we're seeing a safe and tolerable profile. We're seeing the ability to improve motor function as assessed by standard measures of Parkinson's progression, like the UPDRS Part 3 off score, as well as the Hauser patient diary, which is a patient-reported outcome. And across both, we're seeing really important signs that what we intend to generate with this program and improving motor function and quality of life are being borne out in this early clinical work. And importantly, we've begun to look at this data over time, where on this slide here, you can see a depiction of worsening in Parkinsonian symptoms on standard of care in yellow. So the higher the score, the worse that you're doing in motor function over time. And at the bottom, you see a depiction of 5 different dose cohorts of a first generation vector construct called PRO7 and axial NTPD, which was optimized to increase the expression of dopamine. And you're seeing a sustained benefit over time with this. And our goal will be to replicate this in larger placebo-controlled clinical trials. So as the next step for this program, we are continuing to go through dose escalation. As of now, our focus is on completion of a GMP manufactured suspension-based manufacturing process for the delivery of clinical trial material, as well as gaining clearance from the IMPD in the United Kingdom to reinitiate our clinical trials with this new material in hand. Our expectation is to have an update on the clinical trial material and the suspension process in the 4th quarter of this year, and in calendar year 2022, drive towards a path back into the clinic. So let me wrap up with this slide that really illustrates the excitement and the momentum and the milestones that we have over the course of the next several months. The near-term milestone in October of 2021 is the delivery of 12-month follow-up data from our low-dose cohort for axial AAV GM1 across a number of different parameters that we'll be evaluating from a safety and outcomes perspective. For axial NTPD in the 4th quarter of this year, manufacturing and qualified first release of clinical trial material using the revised suspension-based process that's expected by the end of this year. And then 2021 is about execution for Tay-Sachs and send-off children, and we'll continue to drive our screening and enrollment efforts for that program. So we have quite a bit of work to do, but I'm really proud of what the team and our collaborators have done over the course of the past several months, and we are very excited about what is to come ahead for the pipeline for the company. Thank you so much for your attention. Thank you again to the meeting organizers for the time and the platform, and have a great meeting.